Hello everyone, Biochemistry channel is here with the topic titled Lipids-III. Don't forget to like the video before you start watch it, my friends. I wish it to be useful. Let's start. Let's look at the contents of this topic. There are two major titles in the topic. Lipids not carrying glycerine. We will discuss Sphingolipids, waxes, terpenes, and sterols as a main subtitles. And the second title of the topic is lipids linked to other classes of compounds. And there are two major content here. Lipoproteins, which are Chylomicrons, VLDL, LDL, and HDL, and the Eicosanoids. Let's start with the lipids not carrying glycerine. And here the major group is Sphingolipids, which there are too many Sphingolipids. And as you see here, one of them, the Sphingomyelin of Sphingophospholipid. And then we will study waxes, terpenes, and Sterols. Yes, let's start with the Sphingolipids. Sphingolipids, another important class of membrane lipids, also have a polar head group and two non-polar tails. But unlike glycerophospholipids, they do not contain glycerol. Glycerol or glycerine is replaced by sphingosine alcohol. So in short, Sphingolipids are sphingosine derivatives. Sphingolipids have important roles as structural and functional components in the cell membranes of animal and plant cells. They are particularly abundant in brain and nervous tissue. In addition, trace amounts are stored in adipose tissue. Sphingolipids especially present in brain and nervous tissue in large amounts, and they form biological recognition areas on cell surfaces. In humans, at least 60 different sphingolipids have been identified in cellular membranes. Many of these are particularly prominent in the plasma membranes of neurons, and some are clearly recognizable sites on the cell surface. And here you see the glycerol. And in lipids carrying glycerol, fatty acids were attached to the three alcohol groups of glycerine. And we did it in the previous topic. Fatty acids are esterified to glycerol. And this is the sphingosine alcohol. And sphingosine alcohol is an 18-carbon alcohol or alcohol carrying an 18 carbon atoms in the backbone of the molecule. And as you see, there was a double bond also, and an amino group at the carbon 2 position, and also two alcoholic groups at carbon 3 and carbon 3. If we omit that part of the molecule, the first three carbons somehow similar the structure is very similar to the glycerol. Instead, the carbon 2, they carry amino group. And the UPAC name of the sphingosine alcohol is 2-amino 4-trans octadecene 1,3-diol. And the first sphingolipids is ceramides. The carbon 1, 2, and 3 carbons of the sphingosine molecule are structurally similar to the three glycerol carbons in glycerolipids. When a fatty acid, and this is a very distinct feature of the sphingolipids, fatty acid is attached to amino group on carbon 2 of sphingosine alcohol by an amide bond, actually acid amide bond. And the resulting compound is ceramide. And structurally similar to a diacylglycerol. And So this means ceramides are formed by acid-amide bonding of saturated or monounsaturated fatty acid with 18 or 26 carbons to the amino group of sphingosine alcohol. And ceramides are the structural parent of all sphingolipids. In other words, we can say the rest of the sphingolipids are the derivatives of ceramides. And here you see the building pattern of a ceramide carrying a sphingosine alcohol, and then fatty acid represents the backbone of the fatty acid attached to the amino group of sphingosine alcohol. So this is a distinct feature because in the glycerol lipids, the fatty acids are esterified to the alcohol groups. But here, the fatty acids don't form bonds with the alcoholic group of sphingosine alcohol. Instead, forms bond with the amino group. And this is acid amide, and the functional group is here the amide group. Ceramides, as I said, form the basic structure of all sphingolipids. And when hydrolyzed, don't forget, they yield 1 mole of sphingosine and 1 mole of fatty acid. And they are abundant in animal and plant tissues. Fatty acids in their structure are generally saturated or monounsaturated, 16, and in general 18, or the over, such as 20 to 24 carbons. And for example, the nervonic acid, which carries 24 carbons and 1 double bond at the carbon 15. Nervonic acid especially rich in brain tissue ceramides. And the word ceramide, comes from the Latin words cera and amide. And cera means in Latin wax. And ceramide is a component of vernix caseosa. Let me try it for you. Vernix and caseosa. The waxy or cheese-like white substance that covers the skin of newborn human infants. Ceramides are involved in apoptosis, which means the programmed cell death, cell growth and differentiation, senescence, cell migration, and cell adhesion. In the epidermis, it is the main component of the stratum corneum. Together with cholesterol and saturated fatty acids, they provide a waterproofing surface. And stratum corneum, which means horny layer, is the outermost layer of the epidermis and consists of dead cells, corneocytes. And this layer consists of 15 to 20 layers of flattened cells without nuclei and cell organelles. And the cytoplasm contains filamentous cavity. And these corneocytes are embedded in a lipid matrix composed of ceramides, cholesterol, and fatty acids. And this layer also creates a barrier for microorganisms. It also prevents excessive water loss by evaporation. And the amount of ceramide and cholesterol in the stratum corneum decreases with aging. Ceramides or their metabolites are thought to be involved in the emergence of pathological conditions such as cancer, neurodegeneration, diabetes mellitus, microbial pathogenesis, obesity, and inflammation. And for the rest of the sphingolipids, don't forget they are the derivatives of ceramides. Sphingomyelins. Sphingomyelins contain phosphocholine or phosphoethanolamine as polar head groups and are therefore classified as phospholipids, together with glycerophospholipids. And the sphingomyelins means they are sphingophospholipid. In that, sphingomyelins resemble phosphatidylcholine in their general properties and three-dimensional structure, with no net charge in the head groups. They are also classified as sphingospholipids and are phospholipids that do not contain a glycerol backbone. Instead, they carry sphingosine alcohol. Sphingomyelins are found in the plasma membranes of animal cells and are particularly prominent in myelin sheath, a membranous sheath that surrounds and insulates the axons of some neurons, hence the name sphingomyelins. And sphingomyelins particularly abound in membranes and in the myelin sheath surrounding the certain nerve cells. Yes, look at the sphingosine alcohol. When we add an R group here, this is the ceramide moiety. If we add an ethanolamine, a phosphoethanolamine or phosphocholine into the alcoholic group at the first carbon of the sphingosine alcohol, then we get the sphingomyelins. Look at here. The molecular structures of the two classes of membrane lipids are very similar. And phosphatidylcholine here, which is a glycerophospholipid, and the sphingomyelin, which is a sphingophospholipid, have similar sizes and physiological properties, but they play different roles in membranes. The types of fatty acids in the structure of sphingomyelins vary according to the location of the sphingomyelin. For example, the fatty acids in the sphingomyelin of the central nervous system are stearic acid, lignoceric acid, and nervonic acid. The fatty acids in the Schiplan sphingomyelin are palmitic acid and lignoceric acid. And in general, sphingomyelins often contain lignoceric acid as fatty acid. Now look at, they have similar shape, but there are some major differences. And so there are two hydrocarbon tails in the phosphatidylcholine, which both provided by fatty acids. But in here, instead, there is a two hydrocarbon tail. One is provided by fatty acids in here with the acid amide bond, and the second tail is the sphingosine alcohol, not the fatty acid. And the second difference is fatty acids attach to glycerol backbone with an ester bond, but in sphingomyelin, only one mole of fatty acid attached to the sphingosine alcohol with an acid amide bond. Yeah, look at here. We have a triglyceride or triacylglycerol, has three nonpolar side chains and three hydroxyl groups, 1, 2, and 3 here, 1, 2, and 3, are esterified with the three fatty acids. And here you see the phosphoacylglycerol or the glycerophospholipid has two nonpolar side chain tails and one ionic head, and two alcoholic group of the glycerol are esterified with fatty acid, and the third one is esterified with a phosphate group, and also this is a phosphoester, because the other side of the phosphate is esterified with another functional molecule carrying an alcohol group. And look at the sphingomyelin, has two nonpolar side chain tails and one ionic head. The sphingomyelin is formed from a sphingosine alcohol. This is the sphingosine alcohol with a 1 mole of fatty acid and again a phosphodiester bond located at the terminal carbon. Glycosphingolipids, or in short, glycolipids. Glycosphingolipids, which occur largely on the outer surface of plasma membrane, And my dear friends, please remember the last slide of the lipids part 2, which represent the ratio of the different kind of phospholipids in the cell membrane. And glycosphingolipids or glycolipids largely on the outer surface of plasma membranes and have one or more sugar residues as head groups directly linked to the hydroxyl group of the sphingosine at carbon 1 of the ceramide. So what I talk about, I said they are the ceramide derivatives. And they do not contain phosphate groups. In short, they are carbohydrate derivatives of ceramides. They do not carry phosphoric acid and the choline in their structure. Instead, they carry hexoses such as galactose, glucose, and or the N-acetyl derivatives such as N-acetyl galactosamine, N-acetyl glucosamine. And even they can carry the sialic acids such as the neuraminic acid. And this is why we call them the glycolipids. They are divided into 4 classes depending on the change of carbohydrates: Cerebrosides, Sulphatides, Globosides, and Gangliosides. And let's start with the Cerebrosides. Cerebrosides have a single ceramide bound sugar. The galactose ones are characteristically found in the plasma membranes of the cell in neuronal tissue, and the glucose ones in the plasma membranes of cells in non-neuronal tissues. So the galactose, the ones carrying galactose, which we call them galactocerebrosides, are abundant in neuronal membranes in the brain and myelin sheaths of nerves. The ones carrying glucose, it is called glucocerebrosides, are found mostly in extraneuronal membranes or non-neuronal tissues. The synthesis of cerebrosides, which is necessary for the development of the nervous system in the offspring, is possible with the presence of these carbohydrates, and especially here, galactose is so significant. And here, this is the building pattern or the building diagram of a cerebroside. And here you see the blue one is the sphingosine alcohol, and the carbon 2 and fatty acid is esterified. Stop. No, not esterified. This is an amide group, acid amide. And they carry a sugar moiety, glucose or galactose. And fatty acids in cerebrosides have 24 carbons, and differentiation occurs according to fatty acid type. And they are similar to Sphingomyelins in structure. The difference is that they contain carbohydrates here instead of phosphocreatine. If the fatty acid is lignoceric acid, it is called kerosene. If it is cerebronic acid, it is called cerebron. If it is nervonic acid, it is called nervon. These are some examples of different kinds of cerebrosides. Sulphatides, another glycosphingolipid. Sulphatides are sulfated galactocerebrosides. They are formed by the attachment of a sulfate residue to the third carbon of galactose in the cerebrosides. So it's a special type of galactocerebroside, sulfated galactocerebrosides, or shortly sulphatides. They are multifunctional molecules. Approximately 25% of the cerebrosides in the brain are sulphatides. As a cell membrane component, it plays a role in protein trafficking, cell aggregation, adhesion, neural plasticity, and the memory. And it is involved in the nervous system, as I said. Also, they are important for the immune system, insulin secretion, blood clotting, and also against infections. And abnormal metabolism is also seen in many metabolic diseases and is thought to be associated with these pathologies such as Alzheimer's, Parkinson's, and diabetes mellitus. As in this formula, you see a sulfatide molecule carrying a sphingosine and a fatty acid here, and at the first carbon, the galactose moiety is seen, but if you omit the sulfate, we call galactocerebroside, but it's a sulfated galactose here, so this is sulfatide or sulfated galactocerebrosides. Also, it's a class of sulfolipids, which means a special glycolipids containing a sulfate group. And sulfatide is synthesized primarily in the Golgi, starting in the endoplasmic reticulum, where it is converted to ceramide, galactocerebroside, and then sulfated to make it sulfatide. Globosides. Globosides are glycosphingolipids containing 2 or more sugars. So they contain 2 or more, usually D-glucose, D-galactose, or N-acetyl D-galactose. Cerebrosides and globosides are sometimes called neutral glycolipids because they are uncharged at pH 7. And globosides are characterized by their specific carbohydrate composition and are found in the membranes of cells, especially in the nervous system. For example, lactosyl ceramide is a globoside containing glucose and galactose. Globosides have been identified with various carbohydrate antigens are involved in the cell-to-cell recognition, cell adhesion, and other cellular processes. And globosides are also involved in various biological processes, including embryonic development and the immune response. And abnormalities in the metabolism or expression of globosides may be associated with certain diseases. It is important to note that globosides are part of a larger family of glycosphingolipids that includes other subclasses such as gangliosides, lactoseramides, and sulfatides. Each of these subclasses has different carbohydrate structures and functions in cell membranes or cellular membranes. The carbohydrate moieties of certain sphingolipids define human blood groups and therefore determine the type of blood that individuals can safely receive in blood transfusion. And glycosphingolipids are the determinants of the blood groups. A ceramide moiety attached to the antigens here, attached to the carbohydrate moieties. And look at the difference between the O antigen A and B antigen. Here we see the only one type of monosaccharide is different. In O antigen there is no galactose, in A there is a galactose. There is no N-acetyl galactosamine attached to the galactose. In A antigen, there is an N-acetyl galactosamine attached to galactose, and in B antigen, a galactose is attached to the galactose seen in here. And the gangliosides, which are the most complex glycosphingolipids, they have oligosaccharides as polar head groups and one or more N-acetylneuramic acid, this is the difference, residual with a sialic acid at their end. So actually this is one type of sialic acid. They are complex sphingolipids containing many sugar units attached to ceramide, and they contain also sialic acids as an N-acetylneuramic acid. In addition to hexose in cerebrosides, they contain a few more molecules of carbohydrates. So they are also called glycosphingolipids containing sialic acid, and the N-acetylneuramic acid is the sialic acid. Gangliosides are concentrated on the outer surface of cells, especially neuronal cells, where they offer recognition points for extracellular molecules or surfaces of neighboring cells. The types and amounts of gangliosides in the plasma membrane vary considerably during embryonic development. There are also relationships with cell growth and differentiation in carcinogenesis. And tumor cells can trigger cell differentiation by synthesizing a different type of gangliosides. Tumor formation induces the synthesis of a new ganglioside complement, and very low concentrations of a particular ganglioside have been found to induce differentiation of cultured neuronal tumor cells. Investigation of the biological roles of various gangliosides remains fertile ground for future research. Here you see the different types of gangliosides, GM3, M2, and M1, depends on the carbohydrate moieties, and also the content of the NANA, which is N-acetyl neuramic acid, and sialic acid is a sialic acid, gives gangliosides a negative charge that distinguishes them from the Globosides at pH 7. In addition to contributing to the structural integrity of the membranes, the Glycosphingolipids, the Cerebrosides and the Gangliosides, are associated with many cellular functions at the cell surface level. They provide antigenic chemical markers for cells. They act as chemical markers that describe the various stages of cell differentiation. They regulate the normal growth pattern of the cells, and cells are allowed to react with other bioactive substances such as bacterial toxins, glycoprotein-forming hormones, interferons, and viruses. And here also you see together the ceramide, the derivatives of ceramide, sphingomyelin, 2-type phosphocholine containing, and the phosphoethanolamine containing one, and one sugar residue cerebroside and ganglioside. If it's galactose and have a sulfate group, which is called the sulphatide, and also we have the Globosides. And my dear friends, we finished the Sphingolipids. Now we will start to the different kind of lipid molecules. And here the one is waxes. I mean biological waxes. And biological waxes are esters of long-chain saturated and unsaturated fatty acids with long-chain monoamide alcohols, or the aliphatic alcohols, which are the aliphatic alcohols are series of a homologous series of organic compounds containing hydroxyl, or one hydroxyl group attached to an alkyl radical. So aliphatic alcohols can be considered derivatives of alkanes in which one or more hydrogen atoms are replaced by hydroxyl groups. And they carry the long chain of both fatty acid and an aliphatic or monovalent alcohols. And the general formula of waxes is exactly like the general formula of simple ester. This is the functional group of ester. And here the R is an alcohol and a long chain alcohol, and the another R is a long chain fatty acid. So in total, the molecule, the wax molecule has a high carbon number, or high number of carbons in the structure. Waxes act as energy stores and water repellents. The bodies of many plants and animals are covered with layers of wax. They are highly hydrophobic molecules. And look at here, a good example, spermaceti wax. 3D structure here, see? And you see there is a huge carbon single bond, which there's a huge two hydrocarbon part, which they combine with an ester bond here. So the R's carry long chain of carbon, which comes from a fatty acid and the aliphatic alcohol. And spermaceti wax is obtained from the sperm whales. And most commonly, the wax contain lauryl alcohol, cetyl alcohol, ceryl and myristic alcohol as aliphatic alcohols, and myristic acid, palmitic acid, cerotic acid, and melicic acid as fatty acids. And spermaceti is a waxy substance found in the head cavities of the sperm whale. Name spermaceti comes from the mistaken identification of the substance as whale semen, but it's not. Spermaceti was highly valued in 18th and 19th centuries for its unique physical properties. It solidifies at a temperature slightly below human body temperature and becomes liquid again slightly above this temperature. And this made it valuable for a variety of applications, notably the production of high-quality candles. The primary use of this wax is the production of candles, and these candles produce a bright, clear flame and emitted very little odor, making them popular for use in homes and churches. The fact that the wax remained solid in cold weather and could be easily molded in the candles contributed to increased demand. And with the decline of whaling industry due to conservation efforts and the development of alternative wax sources and lightering technologies, the use of sperm whale has declined significantly. And today, alternatives such as paraffin wax and other synthetic materials are widely used in candle production. It should be noted that sperm whales are protected by international regulations, and whaling for sperm whales is banned in many countries, such as Australia, New Zealand, EU countries. And you may check the International Whaling Commission for the recent news information or regulation. And the historical significance of the sperm whale highlights the complex relationship between humans and marine resources and the economic drivers behind specific industries. The melting point of waxes is generally higher than that of triglycerides. They are almost insoluble in water and very well soluble in organic solvents. Waxes are widely found in nature. It is found in the secretion of insects as a protective layer on the skin, hair, and feathers of animals, and the leaves, fruits, and bark of plants. In plankton, the free-swimming microorganisms at the bottom of the food chain for marine animals, waxes are the principal storage form of metabolic fuel. And as we discussed the spermaceti in the previous slides, remember the biggest part carrying an alkane chain, which make them a metabolic fuel. So this means the molecule can undergo the combustion reaction, as in the alkanes. Some skin glands of vertebrates secrete wax to protect hair and skin and keep it supple, slippery, and water-free. Birds, especially waterfowl, secrete wax from their preen glands to keep their feathers water-repellent. And the glossal leaves of holly, rhododendrons, poison ivy, and many tropical plants are covered with a thick layer of wax, which prevents excessive evaporation of water and protects against parasites. Biological waxes find various applications in pharmaceutical, cosmetic, and other industries. Let's talk about some important waxes, and the one is beeswax, in the secretion of the bee. And the beeswax is a mixture of esters of palmitic acid with carbon 26 and carbon 34 fatty alcohols. And bivax is largely composed of myristate and some esters of cerebric acid and a small amount of hydrocarbon. And it melts at 62 or 65 degrees Celsius. And it is used to make shoe polish, candles, and wax paper. Carnivore wax. It's a vegetable wax found in the fibers of Brazil palm, and its main ingredient is myristyl ceratate. It melts at 80 to 87 degrees Celsius, and it's used in polishing, shoe polish, floor polish, varnish, candle, and wax paper. And we have already talked about the spermaceti wax and the lanolin. It's a fatty acid ester of lanosterol. It contains free and esterified cholesterol too. It forms a protective layer on wool fibers and is a wax rather than an oil. It has a very complex structure. Lanolin has the ability to absorb and retain large amounts of water without melting itself. It is therefore used in the preparation of ointments and various cosmetic products. A new title, terpenes. Compounds formed by linking 5-carbon isoprene units are called terpenes. The sterols, fat-soluble vitamins, quinones, and dolichols of all eukaryotes are synthesized from these simple 5-carbon isoprene subunits. And here is the isoprene unit having a 5-carbon, and the UPAC name of this molecule is, as you can easily name it, 2-methyl, as you see here, 2-methyl, and 1,3 here, 1,3-butadiene, butadiene. So this is the UPAC name of isoprene unit. And as you see, there is a conjugated double bonds. And also look at the isoprene unit. 2 isoprene unit forms a 1 terpene. It's called monoterpene. And they are conjugated double bonds in the molecule. In other words, there is a single bond between 2 double bonds. Thus, they have high reaction ability. And the phenomenon of molecules of the same substance combining with each other is called polymerization. So this means the terpenes are the polymers of isoprene unit. Most terpenes are hydrocarbons, others are alcohols, ethers, aldehydes, ketones, and acids. So there are numerous different types of terpenes. Most of them have a pleasant odor, and some of the terpenes are used in perfumes, flowering, and medicine. And here's important ones: the lycopene, carotene, vitamin A, and the scopolamine are some examples of biologically important terpenes, and especially under this title, I will talk about the carotene and the scopolamine. And in real, the vitamin A is a topic of Biochemistry-II course. The most important group of terpenes are carotenoids, and carotenoids carrying a hydrocarbon ring are called carotenes. So there are many carotenoids, and the ones having hydroaromatic rings are called the carotenes. And they are light yellow to red violet in color. Some of them are acyclic, which means they are in ring structure, while others carry hydroaromatic rings, especially at the both ends of the chain. We call them carotenes. And the substances that give many natural oils their yellow color are carotenoids. Since they carry double bonds, They are easily and rapidly oxidized by the oxygen of the air and ultraviolet rays. And let's look at the different type of carotenoids. And the one is aliphatic carotenoids. And there were two here, lycopene, an aliphatic carotenoid of the tomato, which gives the red color. The squalene, I want to put a star here, formed by the polymerization of 6 isoprene units. It occurs as an intermediate in the cholesterol synthesis in animals and humans. This means squalene is the precursor of sterols, which I mean the cholesterol, and actually the cholesterol, and steroid hormones and vitamin D and the bile acids are the derivatives of cholesterol. And we know the cholesterol is synthesized from squalene, so squalene is the precursor of all of them. Aliphatic carotenoids is alcohol group, and here's a phenol. It's found in the green plants depending on the chlorophyll. Aliphatic carotenoids with carboxyl groups, and here the example is alpha-carositine, consists of 4 isoprene units, and it's the substance that gives bile its yellow color. And the carotenes, the most important one of the major groups belonging to the carotenoids, they carry hydroaromatic rings called the ion rings. And the rings are located at the 2 ends of 4 isoprene molecules. And there are 3 types of ion rings: the alpha ion, beta, and the pseudo ion rings. And depending on the type of the rings, there are 3 types of carotenes: alpha, beta, and gamma carotene. Yes, you see the formulas of the ion rings, and as you see, the alpha and beta are real cyclic molecules, which they only differ in the position of the double bond. But the pseudo is not a complete ring, and there are 2 double bonds. And all are the derivatives of the benzene ring. As I said, hydroaromatic, which means the hydrogenated benzene ring. And as I said, there are 3 different types of carotenes. The alpha carotene consists of an alpha ion plus 4 isoprene units and the beta ion ring. And the 4 isoprene and the beta ion ring is same with the other one. So this is that pattern is same for all alpha carotenes. They only differ in the type of the second ion ring. And when the molecule cut into 2 equal pieces, it gives 1 molecule of vitamin A, because 1 molecule of vitamin A contains a beta ion ring and the 2 isoprene units. And the beta carotene, which gives 2 molecules of vitamin A, because the ion rings, both ion rings are the beta ion. So when divided into 2, which means 1 beta ion and 2 isoprene, and the second piece will also be this 2 isoprene unit and the beta ion, and thus it will give 2 molecules of vitamin A. So this is most valuable form of carotene. And in the gamma carotene, it consists of beta ion plus 4 isoprene units and a pseudo ion ring. Due to 1 ion ring, it gives 1 molecule of vitamin A. And this difference is so important because, for example, if you consume a feed containing alpha carotene, assume you got 100 carotene, it will give you 100 molecules of vitamin A. But when you consume a beta carotene, such as when you get a 100 of beta carotene, this molecule will give you 200 molecules of vitamin A. But this is why this is most valuable. And here you see the beta carotene having 2 beta ion rings and the 2 isoprene actually 4 isoprene units, and when we cut the molecule into 2 equal pieces, it will give 2 moles of vitamin A. And this happens in our body with the enzyme beta-carotene deoxygenase. And beta-carotene is a type of carotenoid, a pigment responsible for the red, orange, and yellow colors in many fruits and vegetables. And it's a precursor of vitamin A, and the nutritional essential for maintaining healthy skin, vision, and immune system. And also we say the vitamin is the vitamin of vision. And some food sources which are rich in beta-carotene: carrots, sweet potatoes, pumpkin, spinach, kale, mango, etc. And remember that the beta-carotene is a fat-soluble compound, and consuming these foods with a small amount of healthy fats can increase absorption. Furthermore, a varied and colorful diet in that includes a variety of fruits and vegetables not only provides beta-carotene, but also a number of other essential nutrients for overall health. And the last carotenoid is carotenoids with an alcohol group in hydroaromatic structure. And the xanthophylls is a dihydroxy-alpha-carotene and cryptokysantine, which also gives 1 molecule of vitamin A, which is found in corn grains and red pepper. Thus, we finish the terpenes. Sterols, another lipid family member. They are very common substances in animal and plant tissues. Since their physiological activity is very high, they are compounds that always attract attention. Sterols are structural lipids found in membranes of most eukaryotic cells. The characteristic structure of this important group of membrane lipids is the steroid core, consisting of 4 fused rings, 3 6-carbon and 1 5-carbon ring, and this is called cytosine ring, or cyclopentanoperhydrophenantran ring. What a complex name, my friends, but simply cytosine ring. Let's look at this steroid ring, and also the name cyclopentanoperhydrophenantran ring tell us something or give us more detail about the structure of this ring system. And the phenantran ring consists of 3 benzene rings, and the latter ring is so important. In this steroid ring we have an A, B, and C ring, and these 3 is the phenantran rings, which are the benzene derivative rings. And if this ring is saturated with hydrogen, the double bond opens and the perhydrophenantran ring occurs. So perhydro means saturation of double bonds with the hydrogen. And if a cyclopentane ring, this is the D ring, is added to the perhydrophenantran ring, thus we have the cyclopentanoperhydrophenantran ring is formed, or shortly cytosine ring. And the numbering and lettering in the steroid ring, which you see the numbers, are very important, because different steroids are formed by the insertion and removal of groups on these carbons. And in many of them, there is a carbon side chain or a substituent at the 7th carbon atom, and especially in the Cerebrosides, at the 3rd carbon, they carry an alcoholic group. The and also the other substituents may happen, such as at the 10 and 13, double bonds between 5 and 6, and the other ones. Sterols carry a cytosine ring and the side chain. All have an alcoholic hydroxyl group at carbon 3. They form 3 groups among themselves. And the ones which we will study here is the zoosterines or the zoosterols, which means they are the sterols of animals. the microsterols, sterols found in yeast and fungi, and the most important member is ergosterol. It turns into ergocalciferol under UV light, and this is the vitamin D2, or the plant vitamin D. And ergocalciferol is not found in animal tissue. However, if the animals can consume ergocalciferol, it has a physiological activity in regulating calcium, blood calcium balance, or the body calcium balance. So the animals can use vitamin D2. And the phytosterols, the sterols are plant origin and have two important members. These are stegmasterol and cytosterol, and they are the precursors of progesterone, which is the gestational hormone. So let's continue on the zoosterols in more. Zoosterols are found only in animal tissues, and its most important member is cholesterol. Cholesterol is found in all animal tissues, in the membranes of many animal cells, and the lipoproteins of blood plasma, and is synthesized in the liver. Actually, the liver synthesizes the body requirements, but also we know almost all cells can synthesize cholesterol. But the most provider of the cholesterol for the body is the liver. But also other tissues can do it. For example, the small intestine can also synthesize cholesterol. But the critical tissue is liver. It is mostly found in the brain, nervous tissue, adrenal glands, and egg yolk. And the important things, there is no plant cholesterol. It's only animal sterol. This is why we call it the zoosterol. And here is the famous formula of cholesterol, the major sterol in animal tissues. And it has an amphipathic feature with the polar head group at the hydroxyl group here, as you see. Oops, sorry. Let me to reveal. Where is it? Where is my cursor? Yes, I found it. As you see here with the third carbon, the third carbon of the A ring, there is an alcoholic group, which is the hydrophilic group, or the polar head group for the cholesterol. And there is a double bond with the fifth and sixth carbon at the B ring of the perhydrophenantone ring. There are two methyl groups that attach at the 10th and 13th carbon atoms, which make the 18th and the 19th carbons of the molecule. And also there is a 8th carbon side chain attached to the cyclopentane ring, attached to the 17th carbon of the cyclopentane styrene ring, to the styrene ring. And so this is the cholesterol. Yes, don't forget this molecular structure. It can be a good question for you. So please study it, memorize. And it's actually so easy. In addition to their role as membrane components, sterols act as precursor for various products with specific biological activities. It is the precursor in the synthesis of bile acid, steroid hormones, including steroid hormones and other steroids. So steroid hormones, reproductive hormones such as testosterone, estradiol, progesterone, and adrenal cortex hormones such as cortisol and aldosterone, bile acids, which are required for the fat digestion, and vitamin D also, which is the vitamin of calcium regulation. The cholesterol is required for the synthesis of vitamin D. And also this is the unique feature of vitamin D. It's the only vitamin that can be synthesized in the body from cholesterol. and its effect also similar like the steroid hormones, that's why some say it's a hormone, not the vitamin. And also about 90% of the available cholesterol in the body is used for the bile acid synthesis. So this means the vast majority of the cholesterol is used for the bile acid synthesis and the rest for the other functions. And don't forget, it's also a member of cell membrane. Steroids, sometimes sterol and the steroid terms are used interchangeable, but they are different things. Steroids are the oxidized derivatives of sterols. They have the sterol core, but lack the alkyl chain attached to the D ring of cholesterol and are more polar than cholesterol. So this means steroids are sterols, but not all sterols are steroids. From where they are produced, steroid hormones move through the bloodstream on protein carriers to target tissues, where they enter cells, bind to highly specific receptor proteins in the nucleus and trigger changes in gene expression and thus metabolism. Bile acids. Bile acids are a family of steroid acids synthesized in the liver and play a crucial role in the digestion, thus absorption of dietary fats. Bile acids are polar derivatives of cholesterol that act as detergents in the intestine and emulsify dietary fats, making them more easily accessible to digestive lipases. So the lipases are the enzymes, that require for the lipid digestion. And bile acids are synthesized in the steroids or sterols from the cholesterol through a series of enzymatic reactions, mainly in the liver. And as I said, the studies have shown that the vast majority of the cholesterol, about 80-85% of the cholesterol, is converted into bile acids by the liver. So, bile acids synthesized in liver. Bile acids are oxyderivatives of cholic acid. So this is the acid obtained from cholesterol, and there are different types of bile acids, such as cholic acid, deoxycholic, glutacholic, deoxycholic acid, and the chenodeoxycholic acid. And bile acids do not exist in the free form. After they are synthesized, conjugated with the amino acid glycine or taurine to form the salts, which are the active water-soluble forms of bile acids. And so this means if the cholic acid is conjugated with the glycine, we call it glycocholic acid, or if taurine, taurocholic acid. And especially in animals, the taurine is the primary amino acid used to conjugate that bile acids. And for cats, taurine is an essential amino acid. And also there is another difference between the glycine and taurine. The glycine is a protein amino acid, but the taurine is not a protein amino acid. It's a non-protein amino acid. And here you see the formula of the bile acids, and as you see, it's very similar to the cholesterol, but there was some modification, and also here the conjugation side. And look at here, there's a carboxylic acid, which make them the organic acids. And also by attaching groups such as the hydroxyl group and that position, and also in this position, forms the different kind of bile acids. And also look at here, which is, as you read, removed by the gut bacteria, converting cholic acid to deoxycholic acid, or chenodeoxycholic acid to litocholic acid. Actually, there are two classes of bile acids, my friends, which are the primary ones. The primary ones secreted by the liver, and the secondary bile acids. And the secondary bile acids are produced by the intestinal microbiota through the metabolism of primary bile acids. And as you see, the cholic acid and the cannadeoxycholic acid are the primary fatty acids, and their metabolism in the intestinal microbiota will yield to the oxycholic acid and the cannadeoxycholic acid. And what are the main properties or functions of the bile acids? So they reduce the surface tension and have emulsifying features. They play an important role in the resorption of fatty acids from the intestines. And by expanding the surfaces of fats, they enable enzymes such as lipase to act better on fats. And due to these properties, bile acids facilitate the absorption of lipids, water-insoluble cholesterol, fat-soluble vitamins, and phosphatides by emulsifying them. And some of the bile in the intestines is absorbed and returns to the liver via the vena porta, and from there passes back to the intestines. And this process is called enterohepatic circulation. Bile acids also play a role in cholesterol homeostasis by facilitating the removal of excess cholesterol from the body. And with this slide, we finish the lipids not carrying glycerine. Yes, lipids link to the other classes of compounds, and first we will discuss the lipoproteins. And as you see here, an illustration of a lipoprotein molecule, the carrier of lipids in the blood plasma, and there are different type of lipoproteins, and the second is the eicosanoids. I just give some basic information about the Lysophosphoglycerides and the Lipopolysaccharides. So my dear friends, focus on the lipoproteins and the eicosanoids under this title. Lipoproteins. Some lipids combine with specific proteins to form lipoproteins. They play an important role in the transport of phospholipids, cholesterol, and triglycerides in plasma. Blood plasma lipoproteins are classified according to the particles of lipids they contain and their density. And there are mainly 4 groups plus 1 intermediate lipoproteins, and they contain 50 to 90% of lipids. So don't forget, there are 4 groups plus 1 intermediate, which makes in total 5 types of lipoproteins. And the proteins in the structure of lipoproteins are called apolipoproteins, or shortly apoproteins. And they are classified as apo-A, B, C, and E. And there are also subfractions of each of them. And the lipoproteins in which these apoproteins are synthesized and in which they are found differ. The protein components of these structures organize the entry and exit of particular lipids to and from specific sites. So in other words, the protein components of the lipoproteins give the identity of the type of lipoprotein, because in each lipoprotein they carry a unique lipoprotein or a unique type of apoproteins. And yes, my dear friends, look at the characterization of plasma lipoproteins. So their density can differ due to 2 features, which the one is the triglyceride or the neutral lipids they carry. And the second one is the protein percentage they carry. And there is then we can make an order of lipoproteins according to their density, ascending or descending order. And so what are they? The chylomicrons or the chylomicrons, the CM. The other one is VLDL, very low density lipoprotein, intermediate density lipoprotein, and the low density lipoprotein. And the final one is the high density lipoprotein. And so, don't confuse about the name very low density protein, lipoprotein, because the lowest density occurs in the chylomicrons, or the chylomicrons are the lipoproteins having the lowest density, not the VLDL. And chylomicrons is synthesized by intestines. And look at the content. They contain, or they carry, the highest percentage of triglycerides compared to the other ones. And the one having the lowest content of triglyceride is the LDL and also HDL. But when we look at the cholesterol content, LDL has the highest percentage. And however, look at the percentage of cholesterol, both ester cholesterol and the free cholesterol in HDL, almost equivalent to the cholesterol content of LDL, but they have different metabolic fate. The LDL carry the cholesterol to store in the peripheral tissues. However, HDL called that stored peripheral cholesterol to metabolize into the further molecules that they are synthesized from cholesterol. This is why the LDL referred as the bad cholesterol, HDL referred as the good cholesterol. And VLDL is synthesized by liver. So this is a packaged or the packed lipid molecules having both phospholipids, triglycerides, and cholesterol, and released from liver for the body needs. And in plasma, when the triglyceride content is released, or when the triglyceride or phospholipid or the other contents are picked up by the cells, the remnants is called IDL. And this is an intermediate molecule between VLDL to LDL. And so the LDL and IDL occurs in the plasma. HDL is synthesized by liver and intestine. And look at the apoprotein fractions. What I said, the apoproteins determines the identity of the plasma lipoproteins. And this is so important because by checking these apoproteins, the cells can recognize which type of lipoprotein is knocking the door. So this means cells carry receptors for these proteins. And compare the apoproteins, and as you see clearly, they are unique. For such as in chylomicrons have only B48 and CEAA, in VLDL B100CE, in IDL B100E, in LDL only B100, and in HDL ApoA, C, and E. So these are the differences of lipoproteins. And this is three-dimensional, a vertical cut. Think about that we made a vertical cut of a lipoprotein molecule, and it carries phospholipids, the free cholesterol, and the polar head. Just locate parallel to the phospholipids, and inside the molecule, inside the lipoprotein, there are the triglycerides and the ester cholesterol, or these are the lipid molecules which are not soluble in water. To carry a substance in a blood plasma, there must be a solubility, or the matter must be soluble in plasma. And triglycerides are not, cholesterol, ester are not. And the phospholipids, there are hydrophobic moieties. So this is why we need a system that can be soluble, but also carry the non-soluble parts inside it. So think about the molecule as a globe, as in the Earth, and so the outer surface of that globe is completely hydrophilic. So they must form they are soluble in blood plasma. But inside the globe, they are the packed, stored, or transported triglycerides and the cholesterol esters. So this is the structure of lipoprotein. They are the carrier proteins. They carry lipids. Eicosanoids. Eiko means 20. So they are the compounds carrying 20 carbon. And remember the fatty acids. We have the arachidic acid and the arachidonic acid, which they are the eicofatty acids. So a question, which one is required to synthesize eicosanoids? And we will give the answer in a short while. Eicosanoids are substances that act only on cells close to the point of synthesis, rather than being transported through the blood to act on cells in other tissues or organs. And these fatty acid derivatives have several dramatic effects on vertebrate tissues. All eicosanoids are derived from arachidonic acid. So this is the answer, not the arachidonic acid. The 220 carbon polyunsaturated fatty acid is a PUFA, from which they derive their common name, eicosanoids. They are molecules that are present in very low concentration in the cell, yet have very strong effects. They show similar effects to the hormones. This is why some say they are hormones, or some say they are tissue hormones, because we know the hormones are synthesized by specialized glands, but eicosanoids are not synthesized from special glands. And then another distinct feature of hormones, they are secreted into the blood and they have a target tissue. And also this is the endocrine effect, but it not happens in eicosanoids, or the mechanism or the effect of eicosanoids are different. They are not synthesized in at once, and they are not sent to target tissues via blood. They show the effect on the cell they are synthesized, or in the neighboring cells, which we call them the autocrine and paracrine effect. And they are synthesized as needed depending on external stimuli and act in the tissues where they are synthesized. They are not stored inside the cell. Eicosanoids are effective in reproduction, inflammation, wound, fever, induction of blood clotting, and regulation of blood pressure. So they have various effects. Yes, the first eicosanoids, prostaglandins, PGs. They are formed from prostanoic acid, which is derivative of arachidonic acid, and has a cyclic structure. So some eicosanoids carry a cyclic structure, and the one is the prostaglandin, and their derivative of prostanoic acid. A 5-carbon cyclic structure is carried by prostaglandins. And they are named after the prostate gland, the tissue from which they were first isolated by Bengt Samuelson and Sune Bergström. Prostaglandins have types such as PGA with acid character, PGB with basic character, PGE extracted with ether, and PGF extracted with phosphate buffer, and subtypes of each type are also defined. And the couple of letters A to I in the prostaglandin symbol indicate the ring type of the molecule. The small letter in the prostaglandin symbol after the capital letter, such as alpha beta, shows the number of unsaturated bonds in the molecule. For the F series, the alpha letter is used to indicate the state of the hydroxyl group at carbon 9. Prostaglandins mediate autocrine and paracrine regulatory functions. Prostaglandins have numerous functions. Some of them give the impression of the opposite of each other. Prostaglandins affect a wide range of cell and tissue function. Different types of prostaglandins on a cell, tissue, or system may have different effects, such as gastrointestinal pain, fever, vascular respiration, and reproduction. Such as prostaglandin F2 alpha is released by stimulation of the uterine oxytocin, causing the corpus luteum to lyse, and prostaglandin F2 alpha and prostaglandin D2 act as a constructor in the smooth muscles of the trachea and bronchi, while E2 and I2 act as reductants. And a footnote, some says the prostaglandin I2, its name is the prostacyclin, and some make it another class of eicosanoids. Another member of eicosanoids is thromboxanes, TX. These are the eicosanoids which are closely related to prostaglandins, and actually they are synthesized via prostaglandins, and they have the same precursor as in arachidonic acid, containing an ether group in a 6 ring, and there are two classes, thromboxane A2 and B2. And thromboxanes are synthesized from arachidonic acid found in platelets. They are effective in the formation of blood clots and in reducing blood flow to the clot site. And these are the formulas, or these are the pathways showing how arachidonic acid, in shortly, to the prostaglandins and thromboxane, especially A2. And the arachidonic acid first converted to prostaglandin H2 an intermediate and derived to the other classes. And here at the point of 1, there was a critical enzyme of this pathway, which is the cyclooxygenase enzyme. And there are some drugs in clinics to use to inhibit this enzyme, which are the non-steroidal anti-inflammatory drugs, NSAIDs, non-steroidal anti-inflammatory drugs. So inhibiting that enzyme, which means to inhibit the synthesis of the prostaglandins and thromboxanes. The third member is Leukotrienes. They are eicosanoids that contain 3 conjugated double bonds in their molecular structure and do not contain a ring structure. They are a strong biological signaling molecule. My friends, as in the thromboxane eicosanoids, they do not contain a ring structure. Leukotrienes can cause contraction of the muscles in the airways of the lungs and ultimately asthmatic crisis. And cysteine types of leukotrienes are known as the slow-acting substance of anaphylaxis, SRSA. Leukotrienes cause local vasodilation, increase capillary permeability, and enhance the effects of chemical mediators that may ultimately cause pain and edema. In other words, they facilitate chemotaxis, inflammation, and allergic reactions. And as I said, the cysteine type 1s are known as a slow-acting substance of anaphylaxis. And overproduction of leukotrienes causes asthma attacks, and leukotriene synthesis is a target of anti-asthmatic drugs such as prednisone, a synthetic cortisone. The strong contraction of lung smooth muscle that occurs during anaphylactic shock is part of a potentially fatal allergic reaction in people hypersensitive to bee sting, penicillin, or other agents. Anaphylaxis is a serious allergic reaction that has a rapid onset and can cause death. It typically causes more than one of the following: itchy rash, swelling of the throat and tongue, shortness of breath, vomiting, dizziness, and low blood pressure. And these symptoms typically occur within minutes to hours. And leukotrienes and other ones are derived from the arachidonic acid, but the enzyme is different, or different pathway. The The COX pathway, the cyclooxygenase pathway, yield thromboxanes and prostaglandins. And leukotrienes are synthesized via lipoxygenase pathway. Lipoxygenase pathway. And we'll discuss in Biochemistry-II course in the metabolism. And there are 4 types, the cysteine ones, which are responsible in the SRSA, the leukotriene C4, D4, E, and F4. And so these are the subtypes. And leukotriene B4, G4, and B5. And C4 and D4 cause smooth muscle contraction and are 1,000 times more effective than histamine in narrowing the lung airways. They also increase fluid leakage from small blood vessels and constrict coronary arteries. And B4 attracts neutrophils and eosinophils to the sites of inflammation. Lipoxins, LX. There are 3 hydroxy derivatives of arachidonic acid. As in the leukotrienes, they are obtained in the lipoxygenase pathway. And 2 types have been identified, A4 and B4. And actually, there are some others, or there are epimers of these ones. Although lipoxins are synthesized in very small quantities, they are powerful inflammatory mediators and play an important role in the resolution of inflammation. So they have role in the resolution of inflammation. And by inhibiting infiltration of the polymorphonuclear leukocytes into the inflammatory zone, vascular permeabilization returns to normal levels. It enhances non-inflammatory migration of mononuclear cells and accelerates resolution by allowing apoptotic polymorph nuclear leukocytes to accumulate in macrophages. Yes, we finished the Eicosanoids. Until now we learned 4 major of them: prostaglandins, thromboxanes, leukotrienes, and the lipoxins. And apart from these, there are also eoxins and resolvins. You can check via the web search databases. Eoxins, another eicosanoid, and resolvins. So, a study for you. And the last ones are Lysophosphoglycerides. The one which is found in the membrane, and they occur by the hydrolytic removal of one of the two fatty acids or the glycerine-phospholipids. They serve as intermediate metabolites, and the Lipopolysaccharides, the bacteria, lipids, bacterial, they are found on the outer membranes of gram-negative bacteria, and it's the main building component, and also known as lipoglycans and endotoxins, and they cause strong immune reactions in animals as well as in humans. And my dear friends, we finished the topic. And the references of the topic, you can obtain one of them to study, to widen your knowledge. The next topic is Amino Acids, Peptides and Proteins-I. We will start with the part 1 of amino acids, peptides and proteins. And please don't forget to follow me on Instagram. This is the Instagram account to follow the updates, and also you can visit my website to reach the documents of all slides, and also you can read many articles about my articles on different subjects. Yes, my dear friends, it's time to say goodbye, but before, please do not forget to subscribe to the channel, like the video, and turn on notifications to be informed about the news. So see you in our next topic. Goodbye and stay safe.